day everyone. Welcome to the course on next generation sequencing technologies, data analysis and applications. We have been discussing variant calling. So, in the last few classes we have discussed methods on how to call variants, different types of variants, and we specifically focused on single nucleotide polymorphisms or SNPs or SNVs and small indels. So, we also tested with a script how to call these SNPs or SNVs and in small indels from the SAM output file through a multi-step process. So, in this class I will just summarize what we did in the last class in the hands on, and then we will talk about visual verification of the variant calls. So, we can actually verify whether the variant calls are actually correct visually through looking at the alignment. There are tools for that, and then we will talk about the VCF file format. So, if you remember right in the last class we got the output in this VCF file format, right. So, we will discuss what this format is, what are the fields that are there, what kind of information you can find in that file. So, these are the keywords that will come across in today's presentation. So, VCF, integrated genome browser and data lines. Okay, so, briefly I will summarize the steps that we used in the last class to call variants using SAM tools and VCF tools. There are other tools, but we use SAM tools and VCF tools to do the variant calling, and here are the steps. I have just listed them from the last class. So, again you do not have to remember, but if you are following this pipeline you probably would have to go step by step. So, one is we first converted this SAM file to a BAM file binary format, and then we sorted this binary format. Okay. And this conversion and sorting, these are required for the variant calling. Okay. And in the third part we did something called local realignment. We use a specific tool to do so, and then followed by that we actually marked duplicates, and again we discussed why duplicates are important, why do you have to mark duplicates, etc. The fifth step we did not do, but this is something you might want to do in cases where you have a lot of SNP data, something called base quality score recalibration. It is a time consuming step, so that is why it is not possible for us to cover this in this class or to do hands on in this class, but we discussed about the idea, right, why we do this. I also mentioned the software that will do this for you. And then final variant call, this is the actual variant call where we identify those variants. Okay, again just a flow chart of the scheme. So, we have this output.sam, right. We use this SAM tools view command with these options, and the ultimate outcome is we convert this SAM to BAM. Okay. And that is why we get this output.bam file. So, I am actually mentioning the actual file names that we use, so that you can follow and go back and see the files yourself. Then we did the sorting using this command SAM tool sort. Again you have the options, the output file name, input file name you have to specify, and this will sort by coordinates, right. So, this is the default sorting. So, it will sort by coordinates or positions of the mappings, and this will give you this sorted out.bam. Now, once we have this sorted out.bam, we also generated this index file. So, we will see why this index file is important. This is important also for visualization. So, sorted out.bam.bai, this we use this function SAM tools index and this command SAM tools index sorted out.bam, right. So, we are creating this index file for this sorted out.bam file. Then we use these two files for realignment using this tool called ABRA2 or ABRA2, right. So, then we use this command specifically, right. Again I mentioned what this command actually means. So, we are actually calling Java here, and then we are limiting the memory usage to 6 GB using this command. Then we are calling the program the version 2.23, and we are specifying the input file name, we are giving the output file name, and then we are also mentioning the reference file name, right, which is the reference sequence. Then the threads, number of threads that you will use to run in the processor in the computer. Then we have also created a temporary directory, right, and that where this log will be generated. Okay, so we got this file sorted out .abr.bam, and then we again sorted by name. Okay, so these are actually necessary because of the tools that we are using. They can take data in certain formats after sorting, etc. So this one we call sorted to out.abr.bam, and this sorting is by name, not just by position. Okay, so that's why this -n option. Now, what we want to do is we want to mark duplicates, right. So, these steps are preparing for marking duplicates. So, sorting by name means we are putting these paired and reads close side by side, right, because names of these pairs would be identical. So, the next step is actually using something called SwayamTools fixmate. Okay. So, this will fill in mate coordinates and also fill the insert size, and we created this file name called fixmate.bam. And then again we sorted after we have marked the mates. So, we then sort by the coordinates, default sorting, SwayamTools sort, and we created this sorted.bam. And this is where we are marking the duplicates. Actually, all those steps before were for preparing for this command. Okay. So, we then use this command SwayamTools mark to -r -s sorted.bam, and we created this file final file.bam. So, the marking is done, right, realignment has been done. We did not do the base quality recalibration as we mentioned. So, what we did next is we then called the variance, okay, and this is where we use this command VCF tools mpilup. So, it is collecting all the data from final file, and then followed by that we have this VCF tools call. So, this is where the variant calling is happening, okay, and we have to mention -ploid 1 because we are working with haploid genome. So, this is a yeast haploid yeast, but in case you are working with diploid genome, this will be 2, and by default this is 2. And then we stored these variant calls in this calls.bcf file. Okay. And we can see these BCF files, and we can also filter out some of these variants using this quality argument, right. We want high quality variants. So, we can use this argument qual greater than equal to 20. And then finally, we stored the results using this BCF tools convert option, and from this calls.bcf file we stored this filtered calls.vcf in VCF format, and this is the final file filtered calls.vcf. And we actually had a look also into that file, what this file contains, etc. The commands or tools that we used, so I will just list them here. SwayamTools view, sort, fixmate, mark 2, and we also use the tools after 2. So, you can go and see the options in the manual page, right, what these options mean that we used, and kind of read about all the other options that are also out there. And similarly, for VCF tools we use these commands mpilup, call, view, convert, etc. So, again all these commands they come with their own options, and you have to go through these options and see which one actually will apply in your case. Again, doing that in the class is not possible. So, what I did, I just collected all those commands and we did the hands on, but I encourage you to explore this a bit more. So, what is remaining now is actually looking at the data and also do a visualization and do a manual check, right, whether the variants that are called whether they are correct or not. Okay. So, for that we will use this tool called Integrated Genome Browser or IGB. It is a free tool you can download and install in your system, and you can load these results and you can visualize these results. Okay. So, that is what we are going to do now, and then we will follow up our discussion on these VCF file formats, right. So, we have created these VCF files, and along the way you also saw one BCF file, right. So, BCF and VCF, what these files look like, what is the specific format, we will discuss that after this. Okay. So, let us now go into the visualization part. Let us run Integrated Genome Browser, and also let us look at the VCF file that we generated. Okay. All right. So, let us move on to the terminal, and so I have just listed the files that we created last time. So, using this command ls -l. So, you will see all these files are around, right. You see this sorted.bam, sorted to out dot abre.bam, etc. All these files are here. The final VCF file would also be here, right. So, let us see, right, we have this. If we go up, we see this filtered calls.vcf. Okay. So, this is a file that contains this variant information. Okay. So, let us have a look at this, right. Yeah, filtered calls.vcf, and I just open the file, right, and we again use this command :/wrap, and we will come to the format in a moment, right, what this information is, right. So, you have some header lines here, which again contains something called format, info, and also BCF tools, etc. We will discuss these in much more detail. The interesting part is here, you can see these are the variants. So, again this is the chromosome ID, right, again this corresponds to some chromosome 1, 2, 3, etc. Then you have this position, right, this is where in this chromosome you see this variant. The ID part, this belongs to ID, this dot means there is no information. Then the reference is C, and alternate means this is the variant that is observed compared to the reference sequence. So, alternate is observed is CCT, okay, and the quality score is 36.4, and they should be above 20, right. We filtered with the 20 cutoff, and it says it is an indel. Okay. So, it is an indel means it is an insertion likely, right. So, because in the reference you have C, and in alternate you have CCT, means you have this insertion here, C, one C and one T, and we should be able to see this in our data. Okay. So, let us have a look at this, and then so on. You have these all these interesting variants around here, and there are a lot of indels, and then there are some SNVs, right. You can see these SNV here, T2A or A2G, and again their positions and the chromosome are given. Okay. So, what we will do, we will now go to integrated genome browser, and we will open up this alignment, and we will see whether we can actually see this variant, okay, and whether it is an artifact of alignment or not. All right. So, let us open integrated genome browser. So, I will just show you, right, here it is, right, this is the site where you can download this integrated genome browser from, and you can install this very easily in your system, and you can just say install IGP, right. So, this will install, and you can download and install in your system. Okay. So, I have installed this here, okay, and we can open this. So, if you go on top, right, on the left side, let us see if we can increase the size a bit. Okay. Anyway, I do not see this option here. So, what we can do is I will just open here. If you will on the top, you will find multiple options. So, the one option that we need is the file option. Okay. So, this file option here we have open file, and you can go to that directory where you have created this alignment and all the sorted alignment, etc., right, and we will load this, let us say this sorted out.bam. Okay. This is a file we created during the variant calling process, right, and why are you choosing this? Because IGB requires an index file, right. So, any BAM file that you use, you also need to have an index file created. Okay. So, you can choose any BAM file, but for that you have to first create an index using the SAMTools index command, and then you can load it here. Okay. So, we have already created an index file for this one. So, let us load this. Okay. Okay, and it has loaded actually, and it says you have to zoom in, and then you have to click on load data. Okay. So, how do you zoom in? Here is up here, this zooming, right. So, you can see we are like going into these positions, right. You can see these positions, right, and you can go to any position, okay, and you can load data here. Okay. On this side you have this load data. Okay. So, what you see now is very interesting. Okay. So, here you see the read mappings. Okay. So, each of these bars, right, so this is one read. Okay. And you have, right, multiple read mapping to one position here. So, we can also zoom in further, and you can now see these reads in much more detail. Okay. And there you see different colors because these are the bases that have been colored differently. Okay. And you might be wondering why this is so sparse, right. For some regions there is no read mapping at all, right. You can see this for some regions. So, here is one region where you see these two reads, but here in on the left side there is no read mapping. So, remember we are working with that small data set, right, where we only took about 25,000 paired end reads. Okay. So, this is kind of expected, 25,000 reads will not be able to cover most of these positions. Okay. All right. So, what we are going to do is let us look at this position where we saw this indel, right. So, what is the position? Again going back to the is the 19,971 in this reference NC 001133. Okay. So, if you go back to IGB, right, on the left hand side you can see these IDs, right, and we can expand this a bit, and you see the first one is this NC 001133. Okay. And it also gives the length of that reference, right. So, we are on the right chromosome, right, but we need to find the right position. Okay. So, which is the 19,991, right. So, let us scroll a bit, right, and see 19,991 is probably, or 970. Okay. So, yeah. So, 971, right. So, as you can see here, right, if we can zoom in, yeah. So, you can zoom in and you can see these bases now, right. So, in these positions, right, you can see 19,971, you have these 3 reads mapping, and each of them are showing these insertions. You can see these signs, right. You can go there, put your mouse, and it will show you. Okay. These are type of insertion. You have CT based insertion. Okay. So, all 3 reads are showing this insertion. Okay. So, this is how we can actually verify, right, if this is correct and if the alignment is also okay, etc. You can also load the reference sequence. You can also load the VCF file here. We will not try to do that. It will take a lot of time because the VCF file can be big. Okay. So, one thing I will show you, right. So, we can zoom out a bit and see the read mapping for these positions, right, and we can actually, what we can do, we can actually load the full mapping that I have actually done, and I can show you how this full mapping would look like, right. So, it will look much more complete along these positions. Okay. So, let us load that. I will load that file for you, and this is the full output I have generated, and I have also sorted. I have created an index.bam.bai. So, I will simply load this. Okay. Now, it has loaded. Okay. It is on top. So, I will just image this one. Okay, and I will zoom in, probably we can, okay, I can remove this one, yeah, and I can zoom in and I can load it. Okay. So, this is the full file, right, full output data, and you can see it will look much more complete here. Okay. So, here is this file which was only part of it, right. Here is the one with all the reads, okay, that I have already aligned, etc., and generated the BAM, and you can see it looks much more complete. Okay. I will probably load a bit more data, and you can see this clearly, right. You have these reads now mapping to almost all positions, multiple reads. Okay. And again in this one, if you look at the same position, right, this 19,971, you see this insertion, CT insertion is there. You can see this clearly. This is also marked for you. So, I will zoom in a bit more, and you can see this CT insertion again. Okay. So, this is how we can verify at least some of these variants that we see in the VCF file, and be sure that okay, we are calling right variants, and these are not artifacts of alignment or something else. All right. So, if this is clear, now we can move on to the VCF file format, and we can discuss what we have in there, okay, and how we can read and what kind of information we get in the VCF file. So, I will first go into the presentation, and then we will again have a look at the VCF file. Okay. So, we have seen two file formats. One is VCF, which is called the variant call format. Okay. So, this is a format that we can read ourselves, okay, and it also has a binary version which is called the BCF format. Okay. So, this is the machine readable format, but we can read it or view the contents using these BCF tools. Okay. So, we have done this in the hands on. So, let us now talk about the VCF files and its contents, so that we can read and understand what we have in there. Okay. So, this is important for the subsequent analysis. So, VCF file is a text file which contains information about variants, and it has SNPs, small indels and SVs, right. So, again depending on the analysis, depending on the sample that you are working with, you will have different types of information. It will contain 3 parts mostly. So, you have one part which is called the meta information lines, then you have one header line, and then you have the data lines. So, you can imagine, right, this is like a SAM file we have discussed, right. SAM file you have header lines, then the alignment lines, and similarly VCF file is also formatted something like this. You have meta information lines, you have one header line, and then you have the data lines. So, here is an example of a VCF file, and in the first part you can see the meta information line in blue. Okay. So, this is what I have highlighted, this is meta information lines. You have lot of information, again we will discuss what this meta information line actually contains. Then you have the data lines. Okay. Here actually this is the data about the variants, again we will discuss what these mean. Actually you see a lot of things there, lot of information, and we will discuss what it means. And finally, you have just one line, this is the header line. This actually contains the header for the data lines. Okay. Again we will discuss this header line. Okay. So, data lines are tab delimited. So, they are tab separated text lines, and each line contains 8 mandatory fields. Okay. And the header line mentions names of these 8 mandatory fields. Okay. So, it is just saying okay, what you have in the data lines. Okay. So, what are these headers that you see is chromosome. So, this is the chromosome number, you see this hash chrome if you notice the example. So, this is the chromosome number, this is the first field. The second is the position, this is the reference position where the variant is present. Okay. Then you have the third one, which is the ID or an identifier. Okay. So, this is something looking at some databases and identifying whether this variant is present in other database and has been given an ID, and this ID is then linked here. Then fourth, you have the reference base, the base that is present in the reference sequence, right. So, this is the reference sequence that is mentioned. You have fifth field is the ALT or alternate field that contains the alternate bases. This is actually the variance that we see in our data. Then 6 is the quality, right. So, this is Phred scale quality score for alt, right, or the alternate base, and this is related to the probability that the call in alternate base is wrong. Okay. So, again very similar to the Phred score that we have talked about, right. Then you have the 7th position, which is the filter, which is the filtering status, and this will mention pass if the variant passes all filters, right. So, we have talked about these different types of filters that you can set when we are doing the variant calling. So, this field will describe okay, whether the variant passes all filters that have been set, and or if there is some filter where the variant is not clear, right. So, the variant does not clear that filter, that will be mentioned. Then you have the info fields. Okay. Again we will discuss what these info fields mean, okay. And this will be also described in the meta information lines. Okay. In the data lines, you have some optional fields also, right. So, these optional fields include format fields. Again, we will discuss what these format fields are in detail, and this information is contained in the meta information line. So, that is why I thought maybe it is better to first discuss the data lines, okay, and describe what you have, and then go to the meta information lines, otherwise it might not be clear. Okay. In the data lines, in the optional fields, you can also have the genotype fields, okay, and you will see these two signs, one for unphased genomes and another is for phased genomes. Okay. We have talked about phasing when you are talking about the variant calling analysis pipeline, okay, and this is again applicable for diploid genomes, right. So, for example, if you are working with human genome data, you will have this phasing information there. Okay. Then you can also have sample information, right. So, you can describe some of these characteristics of these formats for each of these samples that you have. Okay. This is again that will be there. Okay. So, here is an example of data lines, as you can see. So, you have first the chromosome number. So, instead of chromosome number, you have some reference ID, again referring to certain contigs or chromosomes. You have positions where the variants are seen, then you have ID, okay, and this ID is related to the some of the database information that is present. Then you have the references, reference base, then you have the alternate base, okay. This actually contains information about the variant that is present, okay. Then you have the quality score, you can see these quality values that are here. Then you have the filter information, again pass means the variant passes all filters that are present, but in one example here you can see this Q10 value, right. So, Q10 probably means this the quality score of the variant should be above 10, but this variant does not pass, right. So, you can see the quality score is below 10, right. So, it is something like 3.4 or so. Then you have some info fields, okay. You can see these NS, DP, AF, A, etc., different characters, and then you have different numbers, okay. And you might be wondering what these actually mean, okay. So, all these terms will be explained in the meta information line, okay. This information will be present and explained in the meta information lines, and once you combine this meta information line with this data line, this will be clear. Okay. So, we will talk about these meta information lines in the subsequent classes, and then you will see this part will be clear. Then you have something called the format field that you can probably not see, only see certain parts for certain variants, like this GT, GQ. Again, these format fields are defined in the meta information lines. Okay. So, this information is present there, and we will use this information to understand what this means. And finally, you have the sample name. Here I am showing just one example sample name. You can have multiple of them, and for each of them you will have this format followed. Okay. So, this format GT, GQ, etc., that you can see partly, this will be actually be repeated for each sample that you have. Okay. So, if you have multiple samples, this information will be given for multiple samples. Okay. So, then we will have the meta information lines we will talk about in the next class, and then you will understand what the data lines actually contain. So, here are the references that we have used for this class. To summarize, we have talked about variant calling using SAM tools and VCF tools in multiple steps, right. So, we have summarized this in this class. We have talked about the hands on that we have done, and we have mentioned the steps and the process commands that we utilized. Okay. So, it might sound very complex, but once you do it yourself, you will see like each of these steps actually do something very important, and there are only few main steps, right, which we have seen. For example, you have the local realignment, you have the duplicate marking, but some of the steps that we have done in the hands on, they are actually preparing the data for those realignment and the final variant calls, etc. Then we have also visualized using the integrated genome browser or IGB. So, we can verify the results that we obtain through the integrated genome browser. So, this is a good way to see, okay, our pipeline is working fine, there is no problem or issue with the pipeline. Then we talked about the VCF files that are generated by the variant calling process, and we talked about 3 parts that are present. So, we have meta information lines, header line and data lines, and in this class we have described the data lines, okay, and also what the header line contain, right. So, header line simply mentions or describes the fields that are present in the data lines. We have not talked about the meta information lines, and in the next class we will discuss the meta information lines, and combining meta information lines with the data lines, you will be able to understand the VCF file completely. Thank you.